Many experiments have been made on transferring the ovary to another part of the body as an autograft or to another animal as a homograft. Such grafts have provided valuable information about the endocrine activity of the ovary. More recently, grafts of this kind have been made to determine the endocrine viability of ovarian tissue, which has been preserved in vitro by being frozen in media containing glycerol. The primary function of the ovary, however, is to produce viable ova, here seen in the fallopian tube. And assessment of this function in a graft is more difficult. At best, grafting causes great loss of oocytes, and to determine the capacity of the surviving ones for normal development, it is necessary to make an orthotopic graft. That is, a graft to the ovarian site, made in such a way as to establish functional connections with the fallopian tube. There have been a number of reports of successful grafts of this kind, but the only substantial body of data comes from Bar Harbor in the United States. The workers there used mice with a genetic marker and sterilized the recipients by surgical removal of the ovaries. This operation, however, is difficult to perform without damage to the site, and the litters obtained by the Bar Harbor workers were often of mixed origin, derived partly from the graft and partly from the remains of the original ovaries. To avoid this difficulty, at the National Institute for Medical Research, we have for some years been developing a different technique, and here is Dr. Parrott to tell you about it.
The essential feature of this new technique is that the recipient animals are sterilized by X-irradiation instead of by surgical removal of the ovaries.
And this is the instrument used for the purpose. It is a Newton-Victor GX10 superficial therapy unit, capable of an output of 140 kilovolts at 5 milliamps. The mice to be sterilized are placed in a cardboard box, which is shallow to prevent them shielding one another during irradiation. As the effects of X-rays upon the ovary are cumulative, the number of oocytes destroyed being proportional to the dose, we reduce the systemic effects of whole-body irradiation by dividing up the required dose into 2 equal parts. The timer is set to give a dose of 300 R. The dose of irradiation necessary for complete sterilization depends on the strain of mice used. The mice are now returned to the animal house. The second dose is given after an interval of at least 2 weeks. The technique can be applied to almost any strain of mice, but in this film a special inbred strain has been used, the G strain obtained from Dr. Faulkner in Edinburgh. Stock animals are agouti with tan bellies. They are heterozygotes, represented by these letters. When interbred, they will produce the following offspring: 1 homozygote, black with tan belly, and 1 homozygote agouti with agouti belly, and 2 more heterozygotes like their parents. It is the 2 homozygotes which we use in our experiments. This is agouti with agouti belly, and this is black with a tan belly. If we now mate a black and tan female with a black and tan male, their offspring will all be black and tan. If we mate an agouti with a agouti belly female with a black and tan male, their offspring will all be heterozygotes, agouti with tan belly. This enables us to demonstrate, without any doubt, the derivation of the offspring. By taking a black and tan female, sterilizing her with X-irradiation, grafting ovaries from an agouti with agouti belly donor, and mating with a black and tan male, we should therefore obtain heterozygous offspring. If the sterilization were not complete, the litter would be mixed with black and tan offspring from the recipient's own ovaries. Now I will show the experimental procedure. The donor mouse, agouti with agouti belly, is killed with chloroform. Now I am going to remove the ovaries. A little spirit to smooth the fur. Abdominal incision. One ovary removed. So. And placed in a petri dish of saline. The other ovary is removed in the same way. The ovary is surrounded by a capsule and by fat, and both of these will have to be removed before grafting into the recipient mouse. Now we come to the recipient. This is anesthetized with avertin, injected intraperitoneally, and within half a minute the animal is ready to be shaved. It is shaved on both sides where incisions are to be made. The areas are wiped clean and antiseptic applied. The animal is laid on its side and an incision is made, first through the skin and then through the abdominal wall. One ovary with its fallopian tube and part of the uterus is exposed. This is one horn of the uterus. This is the fallopian tube. And this the sterilized ovary. A tiny slit now has to be made in the capsule surrounding the ovary. There. The dissected ovary from the donor, or part of the ovary if it is a large one from an adult, is now slipped through the slit in the capsule and gently pushed past the sterilized ovary. This helps to keep it in place. Care is taken not to touch the fallopian tube, and any hemorrhage or damage often associated with surgical removal of the recipient's ovary from within the capsule is completely avoided. Here you see the capsule clearly. The ovary and uterus are now replaced in the abdominal cavity. Then the abdominal wall is sutured, and finally the skin. The whole procedure is now repeated for the other ovary. When the mouse has recovered from the anesthetic, it is returned to the animal house. Here is a section of a normal young adult mouse ovary. There are many ova and developing follicles. And this is a mouse ovary several weeks after irradiation, seen at a higher magnification. There are no ova or developing follicles, only luteal cells from old corpora lutea. Two weeks later, after the incisions have healed on the grafted animals, the grafts will have had time to become established. The black and tan female is paired with this black and tan male. She mates and eventually becomes pregnant. This is a section of an orthotopic ovarian graft from another pregnant animal. Alongside is the sterilized ovary, still showing its connections to the hilus. Note the corpora lutea of pregnancy and many ova in the graft. This is the grafted female with her litter. And here she is in close-up. Note the white patches on her back where she was shaved for the grafting operation. After X-irradiation, all new hair is unpigmented. With her is the black and tan male. And now one of her offspring, which is agouti in colour and therefore derived from the graft. If any of the litter had been derived from her own ovaries, they would have been black and tan like both parents. This technique has been carried out successfully in 6 other different strains of mice. An average of 25 mice of each strain was treated, and the results are expressed proportionally. Here is the proportion of established grafts. The first 3 are inbred strains, and the proportion of takes is high. The last 3 are outbred strains, and the proportion of established grafts is lower. The proportion of fertile grafts is represented by the blue columns, and the difference between the inbred and outbred strains is even more marked. Although there is no genetic identification of young in these strains, such as we had with the G strain, we are certain that the young were derived from grafts because of the regularity of sterilization by X-irradiation. One application of the grafting technique is the investigation of the fertility of ovarian tissue, which has been stored at -79°C, the temperature of solid CO2. The ovarian tissue is kept in ampoules in a special medium containing glycerol. Successful grafts have been obtained, and here is a section of a graft which was stored at 6 weeks before grafting. As you see, it looks very similar to this section of fresh graft. That was the sterilized ovary, and this is the graft. More work is necessary, however, to increase the number of takes and the numbers of litters from the grafts. While we were developing the technique on mice, Adams and his co-workers in America reported that skin homografts could be exchanged successfully between different strains of Syrian hamsters, including an albino strain. These have proved very suitable for our purposes. Hamsters are very sensitive to whole-body irradiation, and so that they stay still during the irradiation, they are anesthetized, and only the ovarian region is exposed to irradiation, the rest of the body being protected with lead shields. 2 doses of irradiation are given, in this case 435 R each, and they are again separated by an interval of at least 2 weeks. Some 2 months from the first dose of irradiation, the sterilized golden hamster is grafted with albino ovaries. The grafting technique is the same as in the mouse. Here you see an ovary from the donor being removed and dissected free from capsule and fat. No normal cross between a golden hamster and an albino male will produce albino young, because albinism is always recessive to the golden eye and coat colour. Here the graft is being slipped into the capsule. But when a grafted female such as this, in which the only active ovarian tissue is from an albino donor, is paired with an albino male, she will always produce albino young. Note that, like the mice, the mother has white patches where she was irradiated. Our first experiments were interesting because the grafts were made prematurely, so that after the first mating some mixed litters appeared. This means at the time of mating there was still some ova left in the recipient's own ovaries, although the graft was also functional. This is a section of the ovary from a hamster which was killed immediately after giving birth to a mixed litter. This is the graft with ova, and this is the female's own ovary, which still has a few ova left. Subsequent litters are almost always completely albino. Here is a section of the orthotopic ovarian graft from a hamster which was killed during her third pregnancy, about 9 weeks after grafting. This is the recipient's own ovary, now completely exhausted of ova and very much reduced in size, and this is the vigorous graft. This new technique of orthotopic ovarian grafting, successfully carried out on mice and hamsters, may find application in many fields of research. In problems connected with the homograft reaction, with the storage of frozen tissue, as has been already mentioned, in genetic research, and in experimental gerontology. It may be also more practical applications in stock breeding will eventually be possible.